Welcome to the pre-lab video for experiment 2, column chromatography. Column chromatography, much like thin layer chromatography that you studied in lab 1, is used to separate the components of a mixture from each other. Column chromatography, however, is used to purify a larger amount of sample, where one can actually isolate and collect the various components of this mixture. Our experimental goals for this week are going to be to identify a mobile phase that will allow the separation of two compounds found in an analgesic tablet. You'll do this using TLC, which you learned in week 1. Then you will separate these active ingredients using your chosen mobile phase on one of these larger scale columns. Before we get into the specifics for our individual experiment, in general, column chromatography uses a glass column packed with silica gel as a stationary phase. This is the same silica gel which coats the TLC plate. It's a white powder, and you'll recall that it's a very polar inorganic solvent. An organic solvent is passed through this packed silica gel as a mobile phase, thus allowing our organic compounds to move through the column. As with TLC, the more polar compounds stick tighter to silica gel, and the less polar compounds will travel more rapidly with the mobile phase. The columns that we use contain a stopper, which can be opened and closed. And in order to keep the silica gel packing within the column, there's typically a plug of cotton covered with a layer of sand. This gives you a nice, even pad of silica gel, which remains inside of the column. Unlike thin layer chromatography, the solvent in column chromatography will flow downwards. It's poured in through the top of your column, and it is allowed through gravity or sometimes applied air pressure to flow out the bottom. So when you think of column chromatography and you want to compare it to TLC, the best way that I can advise you to think about it is as an upside down TLC plate. So let's take an example. Let's say we have the mixture here where we have both the blue and the yellow compounds shown. The blue spot is lower on a TLC plate, so we know that it is more polar. It is absorbing to the silica gel surface tighter. The yellow spot at the top is thus less polar. I'm going to outline it in pink so that you can see it, and travels up a TLC plate faster. If we were to obtain a large amount of this mixture and we were to separate it using column chromatography, we would load our sample mixture onto the top of the column. And as we allow the mobile phase to go through the column, what you'll notice is the less polar yellow compound spends more time with the mobile phase and ideally will elute from the column first. What we strive for is a complete separation where we completely collect the yellow compound before the more polar blue compound begins to elute from the column. In order to run the separation we're going to carry out today, we first have to decide what a good mobile phase for our mixture is going to be. So the analgesic pill you're going to be given contains two active ingredients, acetaminophen and caffeine. So in pairs with your partner, the first thing you're going to do is test out a potential mobile phase to achieve separation of these two compounds. Every pair will be assigned a different mobile phase, and we'll share the data at the board to identify the solvents that seem reasonable to both isolate the material off of the column and to isolate it as two pure components. You'll recall from lab in the past week that caffeine was one of our more polar active ingredients, and acetaminophen had a higher RF value. If we want to be sure that we can separate them in a given mobile phase, we can also have a reference spot that contains a 1-to-1 mixture of the two ingredients together. So when we look at our data as a class, what we're going to be looking for in a good mobile phase is that the two dots actually do separate from each other. We want the less polar acetaminophen, which will come off the column first, to have an RF that's at least 0.3 up the plate. It could even be about halfway up the plate. This tells us that we'll be able to, in a reasonable time frame, have it elute from the column, that it's not stuck to the silica gel. For the lower spot, it's okay if the RF is a little bit lower. We can always increase the polarity of our solvent when we're running our column if it's sticking too much. What's most important is that there is a nice amount of separation between the two spots. Your lab manual mentions that you want at least an RF difference of 0.2 between the spots you're trying to isolate. So once we as a group have identified some viable mobile phases, and there can be more than one mobile phase that seems like it will give us good separation, and if that's so, you can choose the one that's more interesting to you. What we're going to have to do is we're going to have to prepare our pill so that we can actually isolate out these active ingredients. So the pill you'll be given will be in a solid form, and you're going to begin by grinding it with a mortar pestle and adding it to a beaker with some ethyl acetate. Ethyl acetate is an organic solvent, so this will allow the organic components of the pill to solubilize. To facilitate this, we are even going to gently heat it on a hot plate within the hood, keeping the beaker covered to prevent splattering and evaporation. Once you have heated this gently, you'll notice that some components of the pill do not dissolve in the organic solvent. Think about why that might be and what those components might serve as a functional role within the pill. We are seeking to separate the acetaminophen and caffeine, which ideally have dissolved into the ethyl acetate. So we're going to carry out a gravity filtration. We'll have a filter paper inside of this small funnel, and we'll pour our mixture through. All of the solid stuff that we don't care about will remain on the filter paper, and our solution will be filtered through. Label this as beaker M. You're going to take just 1 milliliter of this solution because that's an appropriate quantity to load onto the size column we have available in lab. And you're going to add it to 1 gram of powdered silica gel, which will be provided for you. This is called a preload, and it's basically allowing all of the acetaminophen and caffeine to absorb onto a silica gel surface that will be added to our column. So to give you an idea of what your hood setup will look like, a little more complicated than our experiment 1, you're going to have your glass column on a ring stand with a clamp. You're going to have your gravity filter with a filter paper, your mortar pestle and hot plate, and you'll want to have a UV lamp handy for when we begin to analyze our TLC results. If we take a close-up look at the columns we use in lab, this is one of our columns. So what you can see here is that the pad of silica gel will be created on top of a layer of sand and cotton. And we use this removable clamp and tube to control the flow of liquid out of the column. So once you have your beaker of silica gel loaded acetaminophen and caffeine, all you have to do is make sure your column is ready to go. So what you need to do is go ahead and make sure that you've got your even layer of sand at the bottom. You don't need too much. And then you're going to pour your chosen eluting solvent into the column not all the way to the top. Leave yourself a little bit of room. You want to make sure that the stopper clamp is closed so that the liquid stays in our glass column. And then we're going to add 9 grams of powdered silica gel. And at this stage, we may gently tap the glass to help that silica settle into a nice even pad. You don't want cracks running through your silica column, and you don't want big pockets of air. When you think about your compounds traveling down, you want them to be able to travel uninterrupted through the silica gel surface for the best separation. So once the silica gel has packed nicely into the mobile phase, you may need to drain if you have a lot of excess liquid on top of the silica pad. You can open up that clamp stopper and it'll allow the liquid to drain right to the top of the white silica gel surface. At this point, you'll put the clamp back on and you're ready to add your powder from beaker M2. So this powder will contain the acetaminophen and caffeine that you're hoping to separate. So now it is starting out. You could think of that as the starting line of the race down the silica gel column. And that's going to be your mixture. So from there, you can place your collection beaker number 1 underneath the column. You can open the stopper and you can begin to allow mobile phase to pour through, hopefully carrying our first less polar compound with it. A couple of notes: you will need to keep refilling the solvent because our column can only hold small volumes of solvent at a time. And you never want to allow the compounds and the silica gel to dry out. You want to keep the column nice and moist. Again, this prevents air cracks from forming. And after you've collected about 10 mils or so in beaker 1, you can switch over to the next, beaker 2. We usually recommend collecting about 5 or 6 beakers and then taking a break and looking at what has come out in each beaker using a TLC analysis. Hopefully you'll see a beautiful separation. So that is the way that we're going to carry out chromatography this week. Once you've learned the ins and outs of chromatography, if you find yourself working in an advanced research lab or an industrial lab, you might find yourself having the luxury of using an automated system. In these more complex systems, there's a solvent reservoir that gets pumped through an automatic pump into this pre-packed column. And these samples are collected into test tubes. And what's really cool about these is oftentimes these instruments have a built-in UV detector, so they will be able to tell you which of your fractions contain material before you even spot them on a TLC. But for today, we're going to do it the old-fashioned way, and we look forward to seeing you in lab this week.